Good afternoon and welcome to all. So we are today starting with a new topic, micrometabolism. We have already covered three topics, 3 units, that is history and scope of microbiology. Second one, morphology of bacteria. Third one, growth and nutrition in bacteria. Next unit is microbial metabolism. In the microbial metabolism, we are going to discuss oxidation and reduction reactions. In the microbial metabolism, it is the means by which a microbe obtains the energy and nutrients, for example, carbon, where it needs to live and reproduce. It is nothing but the different methods by which the microbe obtain the energy and nutrients. The microbes use many different types of metabolic strategies, and species can often be differentiated from each other based on this metabolic characteristics. So based on this metabolic characteristics, microbes can be differentiated. The specific metabolic properties of a microbe are the major factors in determining that microbe's ecological niche and often allow for that microbe to be useful in industrial processes or responsible for biogeochemical cycles. Now the types. All the microbial metabolism can be arranged according to the three principles. First one, how the organism obtains carbon for synthesizing its cell mass. It can be mainly through autotrophic metabolism, that is, carbon is obtained from carbon dioxide. Heterotrophic metabolism, the carbon is obtained from organic compounds. Then mixotrophic metabolism, in which the carbon is obtained from both, the name itself, both from organic compounds and by fixing carbon dioxide. Now second one, how the organism obtains a reducing equivalents such as hydrogen atoms or electrons. It can be in this organism obtaining this reducing equivalents used either in energy conservation or in biosynthetic reactions, and those reducing equivalents are used for energy conservation or biosynthetic reactions. And it can be either through lithotropic, that is, reducing equivalents are obtained from inorganic compounds, and also from organotropic, means in which reducing equivalents can be obtained from organic compounds. Third principle, how the organism obtain energy for living and growing. It can be either through phototropic, that means energy obtained from light, chemotropic, energy obtained from external chemical compounds, etc. In practice, these terms are almost freely combined. Typical examples are given. Chemolithoautotrophs, which means they obtain energy from the oxidation of inorganic compounds and carbon, from the fixation of carbon dioxide. Example, nitrifying bacteria, then sulfur oxidizing bacteria, iron oxidizing bacteria, null gas bacteria. These are the examples for chemolithoautotrophs. Next is photolithoautotrophs, in which the energy is obtained from the light. That means photo means light, and carbon from the fixation of carbon dioxide using the reducing equivalents from the inorganic compounds. For example, cyanobacteria, in which water is used as a reducing equivalent, which acts in which hydrogen donor acts as a reducing equivalent. Next is chlorobiaceae, then chromataceae, the hydrogen sulfide as hydrogen donor. Examples. Then another example is chlorofluxes, in which hydrogen is used as a reducing equivalent. Next is about chemolithoheterotrophs. Chemolithoheterotrophs, they obtain energy from the oxidation of inorganic compounds, but they cannot fix carbon dioxide. These are chemolithoheterotrophs. Examples are some thiobacillus, some Beggiatova, and some Nitrobacter species, Vollinella, some Nalgas bacteria, etc. and also some sulfur-reducing bacteria. Next is chemoorganoheterotrophs. They obtain energy, that is carbon and hydrogen, for biosynthetic reactions from the organic compounds. Example, most bacteria, that is Escherichia coli, Bacillus species, Actinobacteria. Next classification is chemoorganoheterotrophs. This is another. They also obtain energy from the carbon. Actually, I had written the same point, sorry. Next is photoorganoheterotrophs. They obtain energy from the light and carbon as well as and reducing equivalents for biosynthetic reactions. And the reducing equivalents for the biosynthetic reactions from organic compounds. Some species are strictly heterotrophic. Many others in this can also fix carbon dioxide and are considered as mixotrophic. For example, Rhodobacter, Rhodopseudomonas, Rhodospirillum, Rhodomicrobium, Rhodocyclus, Helicobacterium, Chloroflexus. They alternatively are called photolithoautotrophy with hydrogen. Next is biochemical reactions. All the living cells depend on these biochemical reactions. For what? For maintaining homeostasis. And all these biochemical reactions in an organism, they are collectively called metabolism. This metabolism is of mainly two types, catabolic and anabolic. Catabolic reactions that break down molecules, generally energy releasing or exergonic. They are considered as energy releasing or exergonic. The next coming is anabolic. Anabolic reactions that build new molecules, generally energy requiring or endergonic. Exergonic reactions provide energy for endergonic reactions also. Next is the metabolic pathway in brief. For example, first one we can discuss exergonic, in which catabolic reactions, energy releasing, they transfer energy from the complex molecule to ATP, in which the simpler molecules such as glucose, amino acids, glycerol and fatty acids are formed and heat is released at the same time, and then this may result in the synthesis of this glucose and amino acid by breaking down, transferring energy. This small molecules, the energy is utilized for the anabolic reaction. So it is called endergonic because the energy is utilized for the to make complex molecules, which means anabolic reactions. In this case also heat is released and the complex molecules such as starch, proteins and lipids are formed by the anabolic process using similar compounds, and the complex molecule is then breaking down to smaller molecules. During this breakdown, energy is released. So that's why exergonic reactions. All the organisms, either prokaryotic or eukaryotic, need to build the molecules they need and find the energy to do so. Next is the most of the metabolic pathway. The biochemical reactions are a part of the series of reactions called metabolic pathway. takes multiple reactions to make an end product. Pathways can be either catabolic or anabolic. Then each reaction is catalyzed by its own enzyme. Next is about enzyme basics. Almost all biochemical reactions are catalyzed by a specific enzyme. The proteins and the enzymes are nothing but the proteins that accelerate rate of reaction without damaging itself, without being changed itself. It is capable of lowering activation energy. Reactions won't occur unless the enzyme that catalyzes the reaction is present and active. The need for the enzyme provides a way to control or regulate biochemical reactions. These are the enzymes which lower the activation energy. The reaction doesn't occur unless the activation energy requirement is met. Activation energy with enzyme and the energy of the reactants, and the activation energy without enzyme. So these are the, due to the presence of enzymes, the activation energy is lower. So no need of rising as much. Enzymes physically bind substrates. This is the enzyme, active site of enzyme. This is the area where substrate bind. Enzyme substrate which forms a complex, and then it may result in the production of products. Again, the enzyme is reverted back to take up other prey. So this is the picture of enzymes. Now the control of enzyme activity. The biochemical reactions can be controlled by making changes in the enzyme activity, which can be influenced in several ways, such as changes in the amount of enzyme or substrate, changes in the temperature, pH or salt, changes in the availability of any necessary cofactors. So these are the important biochemical reactions which can be controlled. So these are the different important enzyme activities. Changes in the amount of enzyme or substrate. So how it can influence whenever there is a change in temperature, pH or salt, etc. Then is the effect of inhibitors. More energy enzyme or more substrate equal to more product can affect enzyme structure, hence its activity. Some enzymes don't work with a non-protein cofactor molecule that bind to the enzyme and reduce their activity. Now these are the factors affecting enzyme activity: temperature, pH and substrate. Whenever the reaction occurs more rapidly as the temperature also rises, but at a particular point it then falls down, and that point is called optimal temperature. At that time the enzyme activity is maximum. Like that it is seen in the pH also, but not in the substrate. The pH affects the charge of R group's protein molecule. Saturation occurs when the saturation is high in. Next is enzyme denaturation. So up to this topic we can discuss enzyme denaturation can be discussed in the next class. Up to this we can wind up the class. Thank you.